A very good morning, students. So we are going to perform our fourth experiment, and the aim of our experiment is to test the and to separate the plant pigments and to study them through paper chromatography, right? So as you can see, I have in my hand a paper which is known as chromatography paper, or we can use Wattman paper number 1 also for this experiment, right? So let's quickly see that what are our requirements for our test today. So aim is clear: to separate the different plant pigments and study them through paper chromatography. So we need pestle motor here, and then because we are going to study plant pigments, so I have taken fresh leaves, and then there is a watch glass we need. Now measuring cylinder, then that is the capillary tube, right? And we need a solvent, that is we are using acetone here. So let's quickly start with our procedure now. What we have to do: first thing, you have to take your fresh leaves, right? You have to take fresh leaves and cut them in small pieces. Okay, so just you can use scissors also, so that it will be easy for you to grind them. If you will take large pieces, then it will take time to crush it, right? So we have to take these small pieces in the pestle motor, and then we have to crush it. So with the help of using pestle motor, I am crushing it. You can see that my sample is partially crushed, so I'm going to use acetone here, right? So add acetone and crush, because we have to make acetone extract, right? I have collected, as you can see in the watch glass, that I have collected sufficient amount of my concentrated leaf extract from the fresh leaves, right? Now we can we have to put this leaf extract with the help of our capillary tube on this Wattman filter paper. Your Wattman filter paper, it should be notched. It should be, you have to cut and form this kind of notch at one corner, and then there should be a mark. I have marked it with the pencil 2 to 3 centimeter above from my notch, right? And this is the place, as you can see this line, this is the place where I am going to put my leaf extract using capillary tube, right? So here I am taking this, as you can see, I am taking this, I have taken this leaf extract in the capillary tube, right? And I'm going to add in the center, right? I'm going to add it in the center of our Wattman filter paper, right? It is very easy to use, so we will use it. And you have to put your extract in the center, just in the center, a thick concentrated. Just let it leave it for a few minutes till it become dry, and we have to repeat this process 6 to 7 times. Again we have to take extract, and then again we have to put it on the same mark for 7 times, right? So I will wait for a few minutes, and then as it is little bit dry, so I'm going to add more, as I have taken it. Now I'm going to add it. So now after putting the, as you can see, thick spot 7 to 6 times, now let it be dry, and then we have to keep it in the acetone. In the beaker I have taken acetone, and I'm going to dip my got a paper in the, as you can see, the solvent is rising, right? As you can see that solvent is rising, right? So let it be stand here for few hours, and till the we have to keep keep it in the position till the 3/4 of the filter paper, because after that only we can observe our pigments. And after few hours we will observe that on the topmost there will be a different kind of pigment, on the middle there will be a different color pigments, right? So different color pigments, they are separated on the basis of the, which is we will calculate them on the basis of our RF value. And with carotene, xanthophylls, chlorophyll A, chlorophyll B, they can be separated out with this experiment. So procedure is clear that first you have to take a concentrated extract, then you have to pour it on the filter chromatography paper, and then we have to keep standing it in the position in the, or dip it in the acetone for the few hours, so that we can separate our plant pigments and then calculate our RF value, right? And then calculate our